Okay, so welcome everyone. The weather's too nice. It's just too nice. So I want to introduce Elizabeth Ryan is Assistant Professor of Toxicology and Nutrition, Department of Environmental, Radiological Health Services, Food Services. You can read the flyer talked about Dr. Ryan's research. It's really, I think, fascinating. You know, the two areas that really, in my mind, are kind of game changers are the impact of the microbiome on human disease and health, and the recognition that certain kinds of foods like rice bran may have very significant long-term health effects. They may be mediated through the microbiome or through other mechanisms. And I was just reading Ghost Map, or read it over the last year, which was the book for the School of Public Health. And I was reading a review of Stephen Johnson, and there's this word consilient that people talked about. And what consilient means is people who are able to take many different disciplines, and because of the perspective of looking at many different disciplines, they look at problems in new and disruptive ways. And I think that's what Elizabeth is really involved with, is it's developing innovative solutions to suit food systems to enhance food security, but it's looking at molecular biology and laboratory animal models and human trials and molecular biology in novel ways, but with a very old food, which is rice bran. So she's going to talk to us today about her work, and the other thing is Elizabeth has been a very active member of our Center for Global Health. We put in a grant together, which unfortunately wasn't funded, but was a great grant, and I look forward to more collaborations in the future.
Thank you so much, Dr. Berman. That was a nice introduction. Okay, and thank you all for coming today. And so I'm going to share a lot of what I'm calling now more of our global One Health research opportunities. Oh, hello, Rebecca. I'm so glad you made it. Sorry. Okay. All right. So here's kind of, when you think about One Health right now, there's been so many different discussions all over, you know, what is One Health? And I'm just going to paint a little picture here about what we're using in our laboratory as a strategy to study rice. And in particular, and so do I have a pointer? Actually, I'm sorry for interrupting there. I don't believe I do.
There is a pointer. It's not always the best, but it's kind of the trigger-looking remote. I think it's over on the right place.
Oh, here we go. I'm just going to use that just so I can help a little bit.
You know what you can do is you can use the mouse to do the pointer on. Let me just see.
I'm sorry. It's fine.
No,
I apologize
here,
for starting
look,
before I knew it. Yeah, I'll
here.
just do it on there.
Just
okay,
do it on here,
that's
and
fine.
then it'll show
Yep.
it on that.
Okay, thanks. So I'm going to tell you a little bit. Our laboratory, as the introduction really said, is that we're doing a lot of different studies. And I wanted to point this out because while we're studying rice, we're working with, you know, animal models, mouse models. We're doing different immune work. We've got cell-based assays and looking at mechanisms of action. We have actually work done now in companion animals. We've done 3 different trials in companion animals so far with weight loss, and companion animals are subjected to many of the similar chronic diseases in particular that humans are getting. We've been doing some work with pigs, in particular germ-free, not-abiotic pigs, and trying to understand what are microbiome-dependent and independent effects of rice bran. Pigs are also a good model for studying the GI tract for young infants, and so we've been focusing on pigs as a model for diarrheal diseases, for instance. And then we also run human clinical trials. And so today, because I can't really tell you about everything, I'll try to do one of our mouse studies as well as talk about some of our human clinical trial work. So why rice, right? Why? Well, rice feeds more than, you know, over half of humanity as a staple food. And I think it's important to note that it's not just about how much rice is produced, but that the people living in major rice-producing areas are suffering particularly from diarrheal diseases, which I'm going to talk about today, as well as we know that chronic diseases are on the rise in the developing world. And this is something we should think about, and I'll constantly go back and forth today between our work in these enteric pathogens and infectious diseases, as well as our work in chronic diseases, in particular cancer. So just if you want to take a look at just a few of the rice varieties that exist, I mean, there's over 100,000 different rice varieties, but I think it's important to appreciate that there's a lot of diversity, right? So there's genetic diversity just within rice itself. So maybe the rice we study here in the U.S. from what we consume is not always going to be applicable to the same effects of the rice in Africa or other parts of Asia. And so what I'm going to be focusing today is mostly on the bran, but I think it's important to know that there are multiple kinds of brown rice, white rice, and then the bran here. And that's one area in which we've been actively focusing on, is finding rice varieties that differ so much, but do they differ in health properties? So, but if you want to take a step now, I'm going to move all the way back, because why even rice? So we know rice feeds most of the world, but is there any evidence for its kind of health benefits beyond just providing calories? And so I think this has been, one of the larger prospective studies that have done to evaluate diet, in particular from staple foods, and have shown that fiber was protective. I mean, this is over 500,000 people that have been followed. Fiber from whole grains and legumes. And that's where, we have a whole other program as well studying beans and legumes. I'm not going to talk too much about that today. But in addition to, this study, which was pretty powerful because it showed fiber intake from whole grains and legumes compared to fruits and vegetables. And that's an important distinction. But there's been others, and this is from the Adventist Health Study, showing just even brown rice intake was protective. So this was kind of a conceptual model to think about how we could start studying foods of different varieties, okay, but I'm going to be focusing on rice. There's primary nutrition. Generally, you think about fats and proteins and carbohydrates and lipids. We talk about them as primary metabolites, but I'm also fascinated by all the secondary metabolites, right, all the phytochemicals that come from our foods, and that the microbiome may be important for not just helping us absorb and digest these foods, but they may be actually contributing to the diversity of chemicals that can be protective or promoting this chronic disease protective phenotype. So how do we even start looking at the microbiome and the effects of foods and looking at health options? I think it's important to take a big look at all the different conditions that are out there that are contributing to stunting in children. And my area of interest particularly has been diarrheal diseases. But one thing I like about this particular chart is the shaded areas kind of talk about where undernutrition in particularly has had some more impact. And that's kind of what this area says. So this idea of diarrheal diseases are a problem, but there's all this undernutrition. I don't know if we've been addressing them together. And so that's the question that we first had was, wow, there's undernutrition. I don't see diarrhea when I see these types of things. We know micronutrient deficiencies are at the heart of overnutrition and undernutrition. But diarrheal diseases, how does that play in both conditions? Okay, because the GI health overall is a concern. So if you go back and just take a look, okay, well, 20% diarrheal diseases here. But I think we need to also think that it's not just that we have an increase in deaths and undernutrition is a problem, but antibiotics are becoming increasingly ineffective, right? So we're having this other piece of resistance. And so what are some opportunities to combat this? And antibiotics are not obviously available everywhere as well. So this provided rationale for a bigger study, and this has been mostly supported by the Gates Foundation, to say, is rice bran an untapped food source or a food ingredient with all these different nutritional properties and phytochemicals that could protect against diarrheal diseases and the undernutrition issue? So here are just a few of the kind of widely known components that have been studied from rice bran. What we've been doing is using metabolomics, so as another approach to get an idea of the full spectrum of small molecules. And we have a few publications looking at that already. But it's not enough because generally these approaches have not been studying the cooked food as we consume it. And that's been our focus, is mostly focusing on the cooked food product as opposed to the raw food or the raw rice grain. So here's just an example of one study that we published where, and this is in collaboration with Jan Leach, who is a researcher at CSU and has been studying rice varieties for a number of agronomic traits. And we got together and said, well, wait a minute, what about all the health traits? So what this is just showing a panel of rice varieties that actually a much larger global rice community has been studying for all these different agronomic traits. And they're also kind of shown here to see, wow, they wanted to pick the diversity that exists, but kind of do a little bit of reductionism, which is what we need to do in research to get a handle on maybe this is a good place to start for differences in health properties. So we just did some metabolomic analysis to say, could there be the same type of variation from an agronomic perspective that could show us in the small molecules? And the answer was yes. So we were confident that this tool could maybe help us as we move forward. But when I went back to the literature, I said, wait a minute, is there any evidence for these brown rice or rice bran compounds having activity? And if you look here, this paper was published in 2000, right? So, I mean, we've got over 14 years now of information that there's already been studies about these compounds. Why haven't they been used? So we dug a little deeper, and we just put a review article out last year that, and this is just a small capture of the number of studies just in cancer. But if you look, rice bran's been widely studied in a number of animal models already and across research groups. But yet still not much was done in intervention standpoint in humans, in children or adults. I mean, they're just absolutely nothing. So we then said, okay, well, what about the mechanisms of action, right? Because we're not ready to jump into people right away, and even though all these animal studies were done, if we don't understand the mechanisms. So they were pretty clear from a cancer protective side that we were looking at antioxidants. We were looking at these different specific proliferative regulation signals. But this bottom half is what I'd like to think about today: immune modulation and mucosal protection. So this is now getting back to our interest in diarrheal diseases, the colon cancer being another area of gut, health that is a concern. Two different kind of spectrums, right? An overweight problem and an undernutrition problem. But how can we start thinking about ways that rice bran may be modulating the immune system as well as protecting the gut mucosa for both conditions? And this is where, it's been a challenge, but if you think in here, look at some of these bacterial enzyme activities. These mucosal protection really could mean protection against enteric pathogens. And so that's what has really moved us forward. But then, I think this one, I don't know how clear that came out. It did. I popped this in there because this is the message, though, right? So I'm going to tell you all about all these health properties of rice. But when you look at the marketing of it, it's really rice, for the people with royal taste, right? White rice is what is sought after as having, the best nutrition you could possibly have. That if you're eating brown rice in most of the world, it's because you don't have access to the polishing that's required, and there's never going to be enough, okay? This is still the thinking. And it's actually become worse and worse. And there's a part of India where my family's from that still does consume brown rice, but really the levels are just much less than I'd ever seen before. So I think there's going to be a lot of room for if this moves forward. And now we have started to see rice bran oil and more brown rice becoming of interest in the developed world. But again, still in most of the developing nations, this is kind of the packaging you'll see. So let's think bagger, because I'm setting us up for wanting to do something in people, but we can appreciate that we're all genetically different in and of ourselves. I've shown you that rice is genetically diverse, and while there's been lots of studies, it's been hard to link kind of all these differences without taking into consideration the microbiome. And this is where both our trials now have been focusing on what are the effects of some of these foods, particularly rice bran in this case, on the microbiome, and how is the microbiome utilizing nutrients from rice bran for bioactivity. So the hypothesis was that rice bran contains bioactive phytochemicals that, when converted by gastrointestinal microbiota, modulate both gut mucosal immunity. So we've looked at a number of those endpoints. And that immunity being important for protection against gut pathogens, but that immunity may also be important for protection against cancer in the gut. Okay, so this is, and again, these fields are typically quite separate. And we want to start thinking about mucosal immunity, though, as connecting them. And then the other kind of piece of our hypothesis is that rice bran may differ in its health properties across these diverse varieties. And this is where we have an opportunity in food security and kind of rice breeding and rice agricultural development. Because if we can identify rice varieties with traits that are of human health importance, we can make sure that they're sustainably integrated or at least available to breeders to maintain, regardless of the variety that you consume. Okay, and that's the piece I don't have time to talk about too much today, but where linkages with agriculture are critical. So here's an example just of if you wanted to take a nutritionist's look at what rice bran contains. Okay, there's a significant amount of fat. Actually, the lipid component of the rice bran is what it's mostly known for. There's also proteins and carbohydrates and many minerals. But we wanted to take a step back and say, well, it's not just these macro micronutrients, but it's also the phytochemicals. And these are the small molecules that we're interested in. And the ones that are in different colors here, and particularly in the reds, the ratio of tocotrienols to tocopherols is unique in rice bran compared to other cereal grains. Another compound that's unique to rice bran is gamma-oryzanol here in blue. And the color coding here is based on immune properties, cancer properties, and then some others that are both that are in yellow. So that's how, and this ferulic acid ester is actually a compound that can be made from the microbiome. But if you consume rice bran, it's really hard to study just one of these components. Or, like I showed you, the micronutrients. You really have to take, the idea that all of this is being taken in and all of it is available to our gut microbiome. And so here is where we have just a summary about genetic selection of our food and how complicated this could be. But we want to make it, realistic. We have individual characteristics that are different. We have to process and prepare our food differently. And so this outer circle of other meal components that we eat it with, could make a difference in the overall ingestion, absorption, and availability of the plant components. And so that's the piece, this outer circle is what we've been paying a lot of attention to prior to designing our clinical trials. And so I'm going to talk to you first about one called Benefit, and it's Beans Bran Enriching Nutritional Eating for Intestinal Health Trial. And so the first goal was how much rice bran can we get adults in and we've moved this more towards a colon cancer focus. And so I'm just going to tell you at the beginning, it was what is the feasibility of feeding 30 grams of rice bran per day to adults for 1 month? Okay, is that possible? And the answer is yes, and we've been able to do this. We started out in healthy adults, and then we now have switched, and we're only doing colon cancer survivors right now. And our first aim was to determine how rice bran intake modulates the gut microbiome. And then the final piece was to take a look at all the stool metabolites that are produced. And that way we could integrate some of that information and not just understand microbiota changes and metabolite changes, but seeing microbial metabolism of rice bran. And that's the piece here. And you can find some more information on this trial. But we're hoping to get out some information on this now from our healthy adults, but we're still enrolling colon cancer survivors. So how do we do it? Here's what we do. We actually designed a bunch of meals and said, wait, we want to control and blind this because nobody really wants to know that they're eating 30 grams of rice bran every day. And we don't want to confound our results from that. So we came up with a series of meals, and we knew this was going to go towards colon cancer survivors, so we tried to think about meals that would be suitable for a wide variety of palates. And there's a control group, there's a rice bran group, and we actually also have a bean group. I'm not going to share that data today. But there's no inclusion of rice bran or brown rice. There are some beans, as you can see, sprinkled in. The dose of the beans is a half a cup a day. Same thing. It's quite a lot more than people are used to consuming. But one meal and one snack per day provides your intervention dose. And then we collect blood, urine, stool, and saliva at the beginning of the study, after 2 weeks, and then after 4 weeks. And this is what's ongoing, and I'll give you some pilot results. So here's what, if you look, C is a control participant, RB is a rice bran participant. The intensity of ferulic acid is shown from our metabolomics data, and then our baseline is the squares here, and 4 weeks is this triangle. So all I wanted to do at first was just to take a look and see if our metabolomics could identify a compound that I know is coming from rice bran, and it be increased in our rice bran participants. But there's ferulic acid could be coming from many other foods. So here's just kind of, your baseline is going to be that natural variation because we didn't have any run-in diet. But you can see that those eating rice bran, after 4 weeks, it was elevated in all of them. And so this was a good indication enough to us at first that we were able to detect compounds coming from rice bran in the stool. But again, ferulic acid is not specific, but it is also one that the microbiome needs to metabolize, to release. And so this was pretty exciting when we first were able to do that. But the next piece was, just in our healthy adults, what kind of shifts are we starting to see? And this is just a look at our rice bran folks. We didn't actually see a lot of change in our control participants, which was reassuring. But in our rice bran folks, because it is such a new ingredient, we did actually see some shifts at 2 weeks that were transient, and then some that stabled out at 4 weeks. And I'm going to just move forward because I want to talk mostly just about specific populations that at the genus level that have shown changes. And one that's been of particular interest to us, and this is just fold change from baseline for all of the individuals consuming rice bran. And in particular, I want to just draw some attention to this bifidobacterium, because we've been looking and interested in specifically how probiotics metabolize rice bran, because, again, probiotics such as bifidobacterium, as well as some lactobacillus species, have been shown to be protective in both the scenarios that I've been talking about, diarrheal diseases as well as in colon cancer. And so could these microbial communities, these probiotics, be part of that, what helps us understand these links in the health of the gut? And so we were excited to see that some were increased. And I'll show you in our mice, actually, we can see lactobacillus numbers increases. But what you have at the beginning, particularly in adults, is going to influence what you can, what rice bran is going to grow on. But overall, this is just a summary from our healthy adults. When we want to switch to the metabolome, right, and understanding what metabolites we can see, we've said, okay, well, we would hypothesize that these are kind of the pathways in which we might see changed and be improved for reducing inflammation in the gut and for protection against colon cancer. And these are very broadly kind of written here. And when you take a little closer look at just metabolomics data, we just want to see how much shift we can get from a baseline, a 2-week, and a 4-week, how much of that is actually transient, as I showed you in the microbiome, versus more of a stable change, or a metabolic change. And this is how we just take a look initially at some metabolomic data, is that what's in the beginning at the baseline sample and what's different at the post-intervention sample. And each one of these dots would be a mass and a retention time. And so then we use a lot of database as well as library matches to identify the compounds. So this is the approach we're using for the fecal metabolomics. And it's pretty standard now. And we're using both GC as well as LCMS. So if you want to take a look at some of the compounds that have changed before and after a rice bran intervention, these are just some, and this is 30 grams per day. So you can see here that beta-cytosterol is also coming from the rice bran itself. The cycloartenol is coming from the rice bran itself. The myoinositol is coming from the rice bran, all of these. And this one at the bottom, I'm going to focus a little bit more on indole-2-carboxylic acid, because it not only has been shown to have anti-inflammatory actions, but it has also been shown to come directly from brown rice. And so there is some evidence. So here, without having a targeted approach, right, we took the stool metabolome and we just started to identify things that were different at the beginning and after intervention, and found actual rice bran compounds in a non-targeted manner, and those that may have some bioactivities. We then started to take a look at some other ones that fall into this bile acids and cholesterol. So I didn't introduce rice bran as something that, but basically it's most known for being able to regulate blood lipids. So the mechanisms, particularly microbiome mediated, have not been well studied. This might be one example that we are outright just promoting the excretion and metabolism of cholesterol into the stool, and as well as mediated through bile acids here. So this is what some other metabolomics has revealed for us as changing before and after rice bran intake. And so as I mentioned, indole-2-carboxylic acid, one of the things, if we wanted to move into a larger population or bigger studies in different areas, is we need some biomarkers, right? This is not like soybeans where you have isoflavones and you can monitor the amount of intake, or you have really key markers of consumption. So identifying compounds that are found in rice bran that we can also detect in stool samples is going to be really important as we move forward and evaluate in larger populations or in children or in collecting stool samples overall. And not needing to use blood. This was the other piece here, is that I wanted to make sure that we had something. We're not to our knowledge yet. We have run urine metabolomics as well, but we haven't identified anything specific to rice bran as we have in stool. The other exciting piece of this is that they're microbial metabolites too. So we can start understanding, would people with different microbiome structures per se metabolize the rice bran differently? And that's something we haven't thought about too much yet, but we need to take into consideration when we think about dietary biomarkers of intake. So here's just what we're calling now, and I put this slide up here not so much to just repeat the metabolites, but now it's really categorizing them as microbial metabolites, right? Usually those that we focused on in a targeted manner were short-chain fatty acids. But now we're trying to say, what is a handful of microbial metabolites that would be indicative of rice bran intake and potentially response, and the response being reducing inflammation in the gut, whether that be important from a diarrheal disease standpoint or a development of colon cancer or even predisposition to obesity. A number of conditions could be considered there. And more is starting to come out in that way. So here's a figure from a book chapter that Erica has recently published. And if you take a look at the top, it's really this idea of we've got infectious and chronic diseases is our problem. And one of the things that we've been trying to address is, okay, well, they're both malnourished conditions as well when you think about obesity being a risk factor for all of these. But there's all these stages, right? I talk about how phytochemicals across varieties might be an option, just food security being a problem, nutrient malabsorption, the integrity of the gut, the inflammation. We have other environmental exposures. I'm a toxicologist kind of by training, moved more into nutrition because I'm interested in where there's some opportunities. But I want to come back to this. But the idea here is this is kind of a cycle that has led to, a lot of different problems, and we haven't really known what to do about it. Where is the opportunity for these staple foods to really alleviate some of these problems along the way? So that's the big picture look at it all again, but then the key is getting at what are the mechanisms? How are we going to do this? So the lab, and this is where we go into animal models, and we need some animal studies. And I've talked to you a little bit about how in humans we saw some kind of probiotic species come up as increasing in the gut following rice bran intake. But now the question is, this is the barrier, should we start looking for some nonspecific IgA? What are some of the different immune cell populations that are changing in the gut or in response to rice bran intake? And so this was kind of the next piece I'm going to talk about how we do an animal study for this. We actually take mice and we would pre-feed them with either a control diet, this is an AIN 93M diet, or a rice bran supplemented diet. And so 10% is what we're aiming for. 10% is what we've been aiming for in our people as well. In adults, we're trying for that to be 30 grams. It seems to be just a little under. But in children, we're aiming for about 15 grams. And we take the tissues out, and we're also looking for serum and fecal lactobacillus numbers because, again, this is something we know from mice to be increased, and IgA. So first I'm going to show you mucosal IgA production. So this is fecal and this is serum from the mice. One thing to note is we started out with these short-term feeding studies, and then we said, you know what, we really need to take this out and cover the full month that we're doing in people. And found that, in fact, it took about 18 days, but we did see in the stool increased nonspecific IgA as well as in the serum. The next question was, what's happening over the same time frame in terms of lactobacillus species in the stool as well? And again, we saw this sustained increase in rice bran compared to the control. And the oscillation that you'll see in the control is kind of these normal native, this is all native gut bacteria, right? We're not feeding any probiotic. And so that's the natural oscillation that you'll see. And we've done some of this ex vivo as well, and rice bran is a very good substrate for many of these probiotics. And so now, again, we're evaluating those metabolites. So we saw this. We saw the bifido, we saw the lacto in the gut, and we said, okay, well, let's infect these mice with what we know to be a known diarrheal pathogen, an enteric pathogen, Salmonella, typhimurium is the strain that we used. And so we, after 1 week of feeding these animals, much like we did in the first study, we decided to infect them. And this is an oral infection dose that would normally, in our pigs, for instance, would produce a diarrheal phenotype, but in mice, they don't actually get diarrhea. So Salmonella fecal shedding is all that we're going to be measuring here. And as you can see, in our rice bran fed mice, we saw a reduction. And this is already published work, and you can take a look at it further. But when we saw this reduction, we thought, wow, maybe, what is the combination of factors, again, that could be contributing to this reduction in the colonization of Salmonella in the gut? So the second experiment, though, we did this just with one rice variety. And so now I just want to tell you, well, how would we choose to do this and understand what in rice bran may be conferring this effect? So we took a look at a number of rice varieties and said, okay, well, we know the lipids are maybe important, right? We hypothesized that the lipid fraction is unique to rice bran and could be important. How could we take a look? And if you look in the black bars here, we just took varieties across the spectrum, but we wanted to cover a range of lipid content. So we took the ones that are in the black bars here. But if you look, even though they have their total lipid content varies so much, their total vitamin E levels don't vary a lot. And so we took a look at a number of different chemicals and targeted analysis of them and found that it's the stoichiometry of all the lipids is what varies between the rice varieties. And it's pretty exciting to take a look at how those will work. But now what's the approach, right? Because I don't want to reduce rice bran down to 1 or 2 chemical compounds. I want to still feed it total. I want to evaluate rice varieties. So we just took different rice varieties. We chemically characterized them, but we fed them whole to the animals. So now you're taking a look, and here's just some more variation. So there's no variation in total vitamin E, but there are differences in gamma oryzanol. There's differences in the phenolic content of these varieties, and now we're going to feed them to mice. Here's some visually of some of the varieties that we chose. We feed them all a control diet. We put them on these different rice bran diets, the same dose, and then we're going to infect them and measure changes in the immune response. This paper we're working on right now, but we do see a number of changes in gamma delta T cells as well as some NK cell populations in the lamina propria. But ultimately, I'm just going to show you today, in the event of time, just the salmonella response. And so, in fact, with this range of lipid content containing varieties, we saw that these 3 varieties did inhibit colonization of salmonella in the gut, and these 3 varieties did not. So I do feel from our animal studies we can identify varietal differences in a health outcome, in salmonella fecal shedding. We don't know, in people yet if that varietal difference will be profound. All of the studies that I showed you that were completed with rice bran previously over the last decade or so, very few people ever pay attention to the variety, if you look at those papers. And this has been one of the areas that we've been kind of charging out there in literature to say, just say what name of the rice, the source of the rice, and where it came from, where it was grown, and this might give us some idea when we're starting to review these studies. So I'm hoping that's starting to make a little bit more attention now. So going back to this diagram, we're taking a look at different immune cell populations. We saw this nonspecific increase in IgA. The idea is not just about if we could promote or increase the colonization and growth of native gut probiotic species, that could be very important in both contexts of protection against diarrhea as well as in some cancer. These interdigitating dendritic cells have been kind of interest as well, and we've shown that they've been upregulated a little bit, and as well as the B cell response. The cytokines, we have some evidence for reducing TNF-alpha as well in our salmonella model. So here's the way we kind of narrowed down and said, what compounds are we going to focus in on? Well, ferulic acid, as I mentioned, is one. Gamma oryzanol is unique to rice. It's actually a sterol and a ferulic acid compound combined. Only microbes have the enzymes that can metabolize some of these components, and that's key for their availability to the gut. And so this is kind of a working model right now. And identifying novel microbial metabolites that might have bioactivity is one piece of this, as well as understanding kind of the mucosal innate as well as adaptive immune responses that are being initiated. So, I just want to kind of highlight up here, and I'm going to take a little bit more time to show you some more kind of global health relevant work, but the microbiome metabolome changes following human rice bran consumption. We believe many of the microbial metabolites to be anti-inflammatory. We're working on understanding how they may be protecting the mucosa and gut integrity. But I think this is something I haven't talked about too much yet. Responders and non-responders, right? We're going to definitely expect to see that in our data. And I think this has been particularly challenging because response to what, right? This level of gut inflammation can vary so much in the system that we're talking about. So maybe a non-responder just needs a higher dose of rice bran to consume, and could our understanding of the microbiome changes help us understand maybe the amount, the dose? Maybe they need to eat it for 6 weeks to see the same changes that we're seeing in another person after 4 weeks. So these are kind of the fluid areas that our research is moving in. We did publish rice varietal differences for the colorectal cancer. We're working on that for the salmonella model, and this is the paper as well for the salmonella infection. Now, what this work has done is led us, as I mentioned, to the pig studies. And this gets to the One Health piece. I like to constantly move between our animal studies, our human studies, and our pig studies, and our dogs, because we learn a lot about the bioavailability and accessibility and kind of actions of these components when we do that. And the pigs, I'd say right now we have human rotavirus infected pigs that are not abiotic, so they have no microbiome, and they get diarrhea from this infection. And when we feed them rice bran at 10% in the diet, just as we have been, we can completely block the diarrhea. And it's just so impressive consistently now. We haven't done it in a treatment context. We have found that rice bran can protect against salmonella post-infection. So that's been really exciting, but we haven't done that yet with the rotavirus. And so this is kind of the area that we're moving into next. And ideally, we should just be able to, I feel confident that we can move into children next too, because this food is safe. The key piece that's holding us up is not so much the safety data of feeding rice bran and maybe understanding its mechanisms of action, but is really the reality on the ground of how rice bran is made available. And
Elizabeth, did you say that you also studied starting the rice bran as a treatment post-infection?
yes, so post-infection, we started that.
And for salmonella, it was helpful?
So in the mice, for salmonella, so basically the mice were given salmonella 2 days after infection. When we would essentially start seeing symptoms, we started feeding the rice bran. And after 6 days, so 4 more days later, we saw reduced fecal shedding still.
And
We used our best variety from our screen to do that study.
and you keep saying 10%.
Yes.
What does that translate in terms of how much brown rice a child or family would have to eat? Since you're giving supplementing with rice bran, what's the...
Yeah, it's not feasible to achieve 10% rice bran with brown rice daily because we're talking about 10% of your rice being the bran, right? So the bran fraction is only 10% of the grain. So you would need to eat like 3 full cups of rice, brown rice per day to achieve that dose. And that's not feasible. Does that help? It's now brown rice consumption has been showing to be protective, right, against colon cancer, in particular colonic polyps. You know, whether or not a consistent consumption of brown rice at smaller amounts over many years is protective, right? Possibly, and very much so. But in terms of the salmonella and any of the enteric pathogen findings, it's all been with rice bran. So, and at these higher doses. And so I threw this slide, I forgot I threw this in here before I get to some of our pictures, but this is a term, environmental enteropathy, that is, you know, growing and growing in the, I'd say, number of people that can discuss it, but it's not just about, you know, bacterial and viral kind of problems, I think, but it's really a subclinical thing. It's being referred to as just the subclinical inflammation. We're getting a little bit more and more interested in it and understanding what some of the biomarkers are and seeing if maybe we could start addressing this condition because it's not full out, you know, diarrheal or problems. But it may also have an involvement of toxicants. And as I mentioned, I'm very interested in understanding the role of how some of these toxicant exposures may also be interplaying with these bacterial and viral, and again, all of this being important. And there's 2 pieces here of kind of maybe some is transient, and is this maybe when you are in environments where you're exposed to this, and then it's more of a transient, and you have a microbiome and a healthy gut system that can handle it, but it's reversed, you know, when your environments change? Or is there some sort of fixed environmental enteropathy that occurs during development that becomes chronic, and even if you move out of this environment, there's no change? And this is just some bigger thinking that's going on in the lab, and what are the opportunities to address this. There are not really animal models or anything like that exist to study environmental enteropathy yet, but that's been a discussion area. Could we start developing some to help better understand this disease or this condition? So let's take a look at some real life issues and what we're dealing with. So here are some pictures from Mali of a collaborator in Mali that is very interested. Rice consumption in Africa is just increasing. Rice production in Africa is increasing, you know, at very high rates. Maybe there's an opportunity as these production scales are increasing in Africa to collect the bran, because it's largely going to cattle or chickens or pigs, right, livestock. Very little, if none, is used for consumption. So here's basically what you're dealing with what looks like a mobile mill, so mills that that are not fixed in location. You can see that the bran is largely on the ground in this. And so if we were to go to Mali and say that, okay, we want to do an intervention trial in children there to look at some of these gut health problems, we first really have a lot of work to do at this stage. And so I think some progress is being made. It is being collected in bins in some cases. But again, it's just, you know, storing it next to maybe corn that has been, exposed to aflatoxins and different molds. You know, rice bran's a good substrate. I don't believe it's a concern as it is. I think, though, if it is housed in conditions with other aflatoxins, it would probably be a good substrate. So these are the things that we need to think about before we could ever assess, I think, its health properties in some of these conditions. But another place, I was just last month in Nepal. You know, same thing. I want to just give you a different look at a mill because that was an outside field, local, mobile mill. We walked into pretty much what I'd call a very small scale but yet mechanized established facility. So here's where people come with all their bags of rice, paddy rice, and you walk in this door here, and the mill operation is all back here. But you can see he actually sells a lot of rice there. The next piece was, if you walk back just in this little tiny room back there, here's basically what you'll see him doing. The other piece is there's just this, maybe you can see scale-wise that it's not much larger than the size of his head, where they're processing all the paddy rice. And then the final piece here is that you separate out and he collects it, but then you can see they collect it for a while and then they stop. So, I mean, the brand's really not, it's the last thing on anybody's radar, because at the end of the day, they want nice, white, clean rice that can be sold and consumed. And so I was just there looking at different sizes of mills and situations of mills and trying to figure out where are some places that we could feasibly collect, because right after we collect this bran, within, I'd say 8 hours, it should be heat stabilized, because I said it's rich in fats. They all oxidize and it goes rancid. And this has been the reason that it's been largely untapped to now. When rice processing and production was at scales where people could consume that bran right away, it was, and it still is in some areas that are very food insecure. But I think the biggest issue is, how can we get this to be collected safely, heat stabilized, and then available for consumption? And this condition may differ depending on the location that you try to do the study. And so focusing a little more attention now. Yes, I want to really characterize all the health benefits, but a little bit more attention, and we need some engineers. Milling technologies have not really advanced here in the U.S. and in the developing world, definitely, but I think there's room to advance them for collection there. So thanks so much for your attention. I just want to make sure I acknowledge Jan Leach and Steve Dow, our other collaborators on the Gates grants that we have for studying rice bran and some mucosal immune properties. Anna McClung and Min Chen are two researchers. One's a breeder and another's a cereal scientist at the USDA. They've been critical to grow our rice bran and provide the bran for our studies. Tiffany Weir is the microbial ecologist that does a lot of our microbiome work. Melissa Wodowick is the dietitian at our nutrition center up at CSU that's worked with us in all our meals, and also getting the dietary recommendations for our patients. My lab, in particular, Erica Borson's here with me. She's critical because she does all of our clinical trial work and going from setting up samples and getting people consented all the way through to collecting their samples and getting them in the lab and processed for analysis. So Erica's been a big player in a lot of these different studies. Andrew Goodyear is a postdoc that just left who'd been working on this. Amy Shefflin's doing a lot of the analysis of the microbiome and metabolome data together. And Dustin Brown's been also very helpful with some sample analysis. Ajay Kumar is the graduate student that did most of the Salmonella infection model. And we use our proteomics and metabolomics facility at CSU. And then Gina Brown is the oncologist for our human clinical trial, and Leckerson for the one that we're doing in children. And so we've had some kind of early grants from the NIH that are still going, and then our as well from the Global Health Challenges. So I'd be happy to have some discussion and some questions. Thank you so much. And I think that's it. Oh, no, I do have a picture of some of the folks involved. Because you can see we're in the kitchen, we're in the greenhouses, we're in Kenya and the rice mills, and it's been fun. And Ginny as well. I'm sorry I didn't mention her, but she's a DVM PhD student, so I've had her for a while to also kind of take a look at the more One Health approaches with this. Okay, and you can see Erica there too. Thank you.